the smallest structures that builds up everything in the body was the atoms, if you remember. Atoms like the carbon, nitrogen, hydrogen, oxygen. Those are very common types of elements that build up a lot of the organic structure that forms our bodies. If you remember, those atoms will form more complex chemical structures. Those are the molecules. Like what molecules? Like sugars, like fat, like protein. All those are complex chemical structures that are actually formed by simple structures, which are the atoms. Those molecules, if you remember, did build up the organelles located in the cells. And organelles like what? Like the cytoplasmic membrane, for example. Like the ribosomes, like the DNA, all those are going to be playing an important role in building up the living cells. And if you remember, the cells here is going to be our smallest living structure, the smallest living building unit of the body. Remember, a collection of cells of the same type is what we've called a tissue. And remember, tissues of different types will build up an organ, like the examples that we mentioned. We did have cells that are gonna be called epithelial cells, for example. They will form together an epithelial tissue. And this epithelial tissue is gonna be sharing along with other tissues, like the muscle tissue, for example, to build the wall of a blood vessel. And the blood vessel here is gonna be considered our organ. So this is the organ level. Multiple tissues building up an organ, and multiple organs, if you remember, they work together to carry on a specific function in the body. We call them the organ system. And we've discussed few examples of organ systems last class. If you remember, the cardiovascular system, the urinary system, the integumentary system, the skeletal system, the muscular system. All those are organ systems that will be building up the organism. And the organisms that we're concerned with in this course is the human. All right, so again, what are the building levels of building organization? Here we started with atoms that will build up molecules, that will form organelles, that will build up cells. Cells of same type will build up tissue. Different tissues will build up an organ. Multiple organs that will be responsible to carry on a specific function will build up an organ system. Multiple organ systems will form an organism. So today what we're concerned with is tissues. This is our main concern in this chapter. In the body, we have four main types of tissues. We have four types of tissues that are going to be building up all the organs in your body. All organs in the body are going to be built by four different types of tissues, and those are the epithelial tissue, or the epithelium, the muscle tissue, connective tissue, and finally the nervous tissue. So again, how many different types of tissues do we have in the body building up all the organs? We have four different types exactly of tissues. Four types of tissues: epithelium, muscle tissue, connective tissue, and nervous tissue. So what's going to be different between those types of tissues? Each one has its specific characteristics. So for example, the nervous tissue is going to be responsible for internal communication. So those are cells that have the ability to send out electrical signal through their limb. Compared to muscle tissues, they will be responsible for movement. So this type of tissue is going to be formed of type of cells that have the ability to contract. And what I mean by contraction is the shortening of the length of the muscle cell. So those cells have the ability to change their length. This is what we mean by contraction. And we do have actually three different types of muscle tissues. We have the skeletal muscle tissue, and this is the one that's going to be located in your skeletal muscles, your biceps, triceps, the muscles that are responsible for facial expression attached to your skin of the face. Those are skeletal muscles, and those are under voluntary control. So you can decide to move your fingers, you're moving skeletal muscles. You can decide to smile, you're contracting skeletal muscles. Another type of muscle tissues that we have in the body, this is going to be the cardiac muscle tissue. And the cardiac muscle tissue is the one that's going to be located in the wall of your heart. And this one is not under voluntary control. So you have no regulation here, no voluntary regulation of the cardiac muscle tissue. So for example, can you decide to speed up your cardiac contraction? What do you think? Can you decide to increase the heart rate? How about the others? What do you think? Can you decide to increase your heart rate? No, I can't. I can't increase my heart rate. I can't reduce the force of contraction of the heart. I can't increase the force of contraction. I can't slow it down. I can't increase it. This is not under voluntary control. All those modifications in the heart rate and the force of contraction are taking place under involuntary control. No voluntary control of the cardiac muscle. A third type of muscle tissue is called the smooth muscle tissue. And the smooth muscle tissue is the type of muscle tissue that's going to be located in the wall of your hollow organs. What I mean by hollow organs, those are all the organs that have cavities inside your body. Like what? Like for example, if we're looking on here to the muscular tubes that will be conducting the food down from the pharynx to the stomach. This is called the esophagus. This is a muscular tube. So this muscular tube is going to have smooth muscles inside that will allow what we call a peristaltic movement to allow the movement of the food down. So what kind of tissue is located in here? It's a hollow organ located in the body. It's gonna be, again, smooth muscle tissue. For you to break down the food in the stomach. So the contraction of the stomach here is gonna be conducted by the effect of the contraction of smooth muscles. It's a whole organ, it has a cavity. Your intestines, small, large intestine, all those are whole organs. Even your blood vessels, will have smooth muscles in their wall, and this will determine the diameter of your vessel. So I can contract the muscles. This will reduce the diameter of my blood vessels. I can relax the smooth muscles in the wall of the blood vessel for me to increase the diameter of the blood vessel. And why would I need to increase or reduce the diameter of the blood vessel in the body? To regulate my blood pressure. So imagine here you have a water hose, and the water hose, the water that is coming out of it is not reaching all the way to the corner of your yard. So what you're gonna do for this water hose? What do you think? What would you do for this water hose in order to get the water reaching a far point in the backyard? You need to squeeze the end, right? And when you squeeze the end, what happens to the pressure of the water coming out? What do you think? So we're talking here about why, what we're gonna do, what you're gonna do when the water coming out of your water hose is not reaching uh, all the way to the back of your backyard. You're gonna squeeze it. You're gonna reduce the diameter of the opening of the water hose. This is the same concept. If you want more blood to travel to your organs, if you're running, so what you're gonna do to your blood vessels? Here, some of the blood vessels need to constrict for you to get the blood reaching those organs. More blood at a high velocity reaching my organs. So you see on here, I can dilate, I can constrict my blood vessels using what type of muscle tissue again is going to be located. It's going to be my smooth muscle tissue. It's a hollow organ. It's an organ that has a cavity inside. What else? Examples of hollow organs that we have, like the gallbladder, for example. For drawing here, the liver is a solid organ. It doesn't perform contraction. Here, it's a solid organ. So we can see on here, on the inferior surface of the liver, we've got a bladder here that will be responsible to collect the bile released from the liver. And this gallbladder, when you get food in your small intestine in here, it needs to contract in order to squeeze the bile to travel to your small intestine. So what kind of muscle tissue do you have in your gallbladder? It's a hollow organ, so it has a cavity inside, so it's going to be smooth muscle. Your urinary bladder, the ureters, the urethra, the uterus, all those are going to be organs with cavities inside. And if I have an organ with a cavity, what kind of muscle tissue will exist in the wall of those organs? It's going to be exact smooth muscle tissue. So another Another type of tissues other than the nervous tissue, which is going to be located in the brain, spinal cord, and nerves, and the muscle tissues is going to be either skeletal, cardiac, or smooth. We have a third type of tissues, which is the epithelium or the epithelial tissue. Epithelial tissue is actually two types. We have the glandular epithelium, and we have the surface epithelium. Glandular epithelium is the one that builds up my glands. So for example, if I'm looking at the salivary glands that are going to be producing the saliva in my oral cavity, those glands are going to be built by clusters of epithelial cells arranged together to form a gland. Glands like, for example, your in the breast, for example, in females, mammary glands. In the wall of my small intestines, I have glands that are responsible to release the intestinal secretions. All those are going to be built up by epithelium. And we have the surface epithelium, and the surface epithelium is the one that's going to be forming the linings and coverings of any organ. So, for example, if I'm looking at the stomach in here, so this stomach has an internal surface. So it will be aligned with epithelium on the inside. And it has an outer surface. So it's going to be covered with epithelium. So again, epithelium here is going to be forming the boundaries between the different environments. It's going to be forming the linings and coverings of all organs. All right, we're going to get to the details in a second, Theodore. All right, so glandular is the one that builds up the glands. Surface is going to be the ones that will form the linings of the organ or the outer covering of the organ. So any organ that has an internal surface is going to be lined with epithelium. And all the organs definitely going to have an outer surface, so they are covered from outside by epithelium. All right. A third type, or our last type of tissues that we're going to discuss in this chapter, is going to be the connective tissue. And the connective tissue, from its name, what is going to be its function? It serves as a connection. It builds up the internal structure to provide support, to protect, and to bind the other types of tissues. So other types of tissues are connected and attached together by connective tissue. This is briefly the different types of tissues. So let's start with our first type of tissue, which is going to be the epithelium. We have two main types that will be determined by location. First one is going to be the glandular epithelium. Second one is the ones that will be forming the coverings, the linings of the organs, and we call it this the surface epithelium. Again, glandular epithelium is the ones that builds up glands. Surface epithelium is the ones that will be forming the external covering and the internal lining of an organ. So what are the characteristics here of the epithelium? Please pay attention here for those 5 characteristics. We're going to rely on those 5 characteristics in our discussion today, and I will be asking many times, referring back to them. All right, so the first characteristic of the epithelium is that cells will have polarity. What do I mean by polarity? I have 2 poles, 2 sides. What this means? If I am an epithelium, so the epithelium being forming coverings or linings of the organ, so it's gonna have one side that is gonna be attached to other tissues, and the other side of the epithelium is gonna be exposed to the outside, exposed to space. So again, if I'm looking at any epithelium, it's gonna have 2 sides. Sides that will be attached to the underlying tissues because I'm forming the outer covering, let's say. So if this is my, so it's not appearing on this. If this is a cell that is forming my outer covering, so this cell has a surface, a side that is exposed to the space, nothing attached to it from one side, and it's gonna be attached to tissues on the other side. So this is what we call by having polarity. I have 2 sides. Unlike a connective tissue that is gonna be connected to one tissue on one side and another tissue on the other side. So I'm connected to tissues from both sides. Epithelium has polarity. One at the apex, and we call this is my apical side, or the free border, or the upper border. So again, what do we call this border on here? It's my free border, or my apical border. The one at the base in here, this is my basal side or lower side, and it's going to be attached to other tissues, not free. This is what I mean again by having polarity. Second characteristic here of the epithelium, the epithelium are composed of closely packed cells that have tight junctions in between. If I'm telling you those epithelial cells are forming the boundary, they are forming the covering or lining of an organ, would you like to have spaces between the cells so anything can pass in or out? What do you think? If you are the one who is designing a tissue, I'm telling you, I want you to design something that will act as a barrier. Would you like to have spaces between the cells to forming this barrier or not? Definitely not. So the cells in the epithelium, are closely attached to one another. You have tight junctions in between the cells, very strong junctions to prevent anything from passing between the cells. So don't want to have something passing between the cells. You want to regulate what's going to be able to pass to the deeper tissue. So for example, your skin, when you get when your skin get exposed to water, let's say you're swimming, why the water does not penetrate to your deeper tissues, why you're not swelling if you are swimming in a pool, why this water doesn't get to the inside penetrating your cells, because simply those cells are closely attached, strongly attached, closely packed together, preventing anything from passing in between the cells. So if you do want to allow water, for example, to pass into your tissues, you would need to allow it through the cell, mainly through the cell, not between the cells. So here, it's a characteristic that makes perfect sense, as this epithelium is acting as a barrier, a surface barrier. It's either a barrier covering the organ from outside or lining the organ from inside. So in both conditions, you don't want to have something passing between the cells. You are acting as a barrier. Another type, another characteristic of the epithelium, which is very important, is that the epithelial tissue is avascular. What I mean by avascular, A means it is without vascular blood vessels. So this is the type of tissues that doesn't contain blood vessels embedded in it. Why do you think you don't want to have blood vessels embedded in your epithelium? Is this something good or bad to not have blood vessels embedded in the epithelium? Good or bad? What do you think? Remember what we're looking at, it's a barrier. A barrier. So a barrier that is exposed to the outside. So if you don't have blood vessels, would you more or less likely to bleed if you get exposed to an irritant or to a trauma? More likely, if you did have blood vessels in the epithelium, at any trauma, you're gonna bleed. So it makes perfect sense that acting as a barrier doesn't you don't want to have blood vessels embedded in this tissue, because being a barrier, forming the covering and lining of the organs, you don't want to have, you are exposed all the time, to trauma. You are forming the covering and lining of the organs, so all the time you are exposed to trauma. So if the epithelium was vascularized, did have blood vessels, I am more likely to bleed. Try to imagine every time you touch your skin, a blood vessel ruptures because the outermost layer, it contains blood vessels, and you're gonna be bleeding every time a person touches his skin. So is it something good or bad to be avascular as epithelium? What do you think? It's it's good, definitely. If I did have blood vessels embedded in my epithelium with very minor trauma, I would have lost blood. But what is the bad side of it? The bad side of it is that those cells are not getting enough nutrients and oxygen on their own. They need to be supported by the underlying connective tissue at the base. So this tissue is the one that contains the blood vessels. Fluids carrying oxygen and nutrients will be diffusing to reach this epithelium, like this. So what is the good thing of not having blood vessels embedded in my epithelium? It's gonna reduce the risk of bleeding, as I am forming a barrier, a surface barrier. So I am less likely here to bleed if I don't have blood vessels. But the bad side is that I can't be independent. I can't rely on my own, on myself. I need to be supported by the underlying connective tissue at the basal site. So every epithelium, every epithelium, it's gonna be supported by the underlying connective tissue. Yeah, it's nourished by the connective tissue. Yes, it's nourished by the connective tissue. So you need to get your oxygen, your nutrients, everything for those cells to survive from the connective tissue. All right, so physically supporting the epithelium, yes, and also it allows the fluids carrying oxygen and nutrients to travel up. All right. So please mark those 2 points down, number 3 and number 4. Also, number 5. So as I am exposed to trauma all the time, would you like those cells to be rapidly dividing or slowly dividing? What do you think? Rapidly dividing. Why? Why? I'm all the time exposed to trauma. And as I get exposed to trauma, what's going to happen? Some of the cells will be injured. And those cells that got injured, they need to be replaced. Exactly. So by other living cells. So the living cells will have a high proliferation rate or high rate of regeneration for me to be able to heal quickly. All right, so again, 5 very important characteristics of the epithelium that we're looking at in here. First one, cells have polarity. What I mean by having polarity, I have an apical side and a basal side. Apical side is not connected to any tissue. It's my free border. And the basal side is the one that will be attached to the underlying tissues. I'm forming a barrier, so the cells need to be closely attached together to not allow anything to pass in between the cells. This epithelium is avascular, it doesn't contain blood vessels, so it needs to be supported by the underlying connective tissue. Those cells are rapidly regenerating for you to have a faster healing process, as this type of tissue is the one that is exposed to the outside. It forms the lining and coverings of your organs. So how do we classify the epithelium? Epithelium that will be forming the linings and coverings of the organs is going to be classified according to, first thing, the number of cell layers that are building this tissue. So if I have one layer of cells, it's going to be classified as a simple epithelium. If I have many cell layers, it's going to be classified as stratified. So what I mean by a simple epithelium is an epithelium that is formed of one cell layer. A stratified epithelium is an epithelium that is formed of more than one cell layer. This is the first way by which we're going to be classifying the epithelium, either simple or stratified. Another way by which we're going to be classifying the epithelium is going to be according to the shape of apical cells. What do I mean by apical? Remember, epithelium has polarity. It has two sides. A side that is going to be pointing towards my tissues. This is my basal side. And the one that is directed towards the space. This is my, at the top, this is my apical side. So the apical cells, the top cell layer, is the ones that we're gonna use to classify the epithelium. All right, so we're gonna only be looking at apical cells, the top cell layer. If the top cells are flattened, flat cells like this, we're gonna call this as my squamous epithelium. Squamous epithelium. If the cells are cube-shaped cells, they are cubes. So the height of the cell is almost equal to the width of the cell. I look like a cube. So we're gonna classify this epithelium. It's a cuboidal epithelium. If I am formed of tall cells, so the height is much greater than the width, we classify this epithelium as columnar epithelium. So again, two factors that allowed us to classify the epithelium. First, according to number of cell layers building up this epithelium. If I have one row of cells, this is classified as simple epithelium. If I have many cell layers, it's going to be classified as a stratified epithelium. Stratified epithelium. The other factors that we're going to use to classify the epithelial tissue is going to be according to the shape of the apical cells. According to the shape of the apical cells, we've got squamous epithelium. This is the type of epithelium that has the outer cell layer, the top cell layer is flattened. If the outer cell layer or the apical cell layer is formed of cube-shaped cells, so this is going to be classified as a cuboidal epithelium. If the top cell layer is formed of tall cells, this is going to be classified as a columnar epithelium. So first thing, in order for us to be able to identify this type of tissue here, what do you think? What kind of tissue are we looking at in here? I see a space first. What can I see? I see space, empty spaces. And those spaces are lined with cells. Lined with cells. So if you have a space lined with cells, so this tells you what? This tells you that you're looking at epithelium. Epithelium is the type of tissues that forms the linings and coverings of the organs. So how did we classify the types of epithelium again? First characteristic according to which we're going to be classifying the types of epithelium is going to be according to the number of cell layers. So we're looking here. How many cell layers can I see on here? Focus on just one part at a time. How many cell layers? Have only one row of cells. So this is going to be classified as what again? If you have one row of cells, it's going to be classified as simple epithelium. Simple epithelium. So let's check, what is the shape of the cell? So when you look at the shape of the cell, how many cells would you need to look at? You need, when you look at the slide, to look at, when you are looking for the shape of the cell, look at only one cell at a time to not get confused. So I will pick one cell, say this one. Or this one at a time. So what do you think? Is this squamous, cuboidal, or columnar? Does this one looks like this, or this? Squamous, right? Flattened cell. So what type of epithelium am I looking at? First, I need to know that this is epithelium. I see a space lined with cells, epithelium. In the type of epithelium that has one row of flat cells. So it's a simple squamous epithelium. Simple squamous epithelium. So let's look at a different one on here. What do you think? What can I see? Please break down what you're looking at in order to make sense. I'm looking at spaces here. See? And those spaces are lined with cells. So we're looking at tubes, many tubes that are located next to one another. And I'm looking at each of them, only one tube at a time. So what I find, I would see that I have a space. I have a space. And this space is gonna be lined with cells. So lined with cells, space lined with cells. This tells me I'm looking at epithelium. How did we classify the epithelium? According to two factors. First is the number of cell layers. How many cell layers do you see on here? Because some of you will look at this diagram, will say, yes, this is one cell layer, and this is another cell layer, or this is 1, 2. So those are not two cell layers. What you're looking at in here is just one cell layer all around lining the tube. So I cannot say, if I'm looking at a tube here, I have one cell on this side, and I have one cell on the other side of the tube. I cannot consider those are two cells lining the tube. The tube is lined just by one cell all around. So you cannot consider those as two cell layers. Why? Because simply you have the space in between. And as we mentioned, epithelium doesn't have spaces between its cells. Cells are closely attached. So I'm looking at how many cell layers here? Just one cell layer lining all around the tube. And what is the shape of the cell? So just look at one cell at a time. Look in here, shape of this cell. Or this cell. What do you think? Is this cell looks like a squamous, cuboidal, or columnar? See, this is the width of the cell, and this is the height of the cell. So they are very, very, very close. The height is almost the same as the width of the cell. Very close. So if I have the height of the cell is the same as the width of the cell or very close to this, it's going to be considered a cuboidal exactly. So what is the type of epithelium that we're looking at in here? I'm looking at a type of epithelium formed of one layer of cube-shaped cells. So what do we call this type of epithelium? It's my simple cuboidal epithelium. Simple cuboidal epithelium. How about this one? So what am I looking at? I'm looking at a tube here, like this, and I did cut the tube into two halves. So I'm looking here at one half of the tube. And I'm looking at the internal lining of the tube. So you have a space. This is a space. This is a lumen of the tube. And this lumen is lined with one cell layer or two cell layers. What do you think? If I see those cells on here and those cells on here, are those two cell layers or one cell layer lining the tube? Just one cell layer lining the tube on both sides. All right. So again, how many layers of cells are lining the tube? All right, we're gonna go for a break in just 2 minutes. For those who have technical issues, just uh, just be patient. We're gonna be back in. So we're gonna have a break in just a minute. All right, so I'm looking at one cell layer lining the tube, but I see actually the two sides of the tube, upper side and lower side. So you see on here, this is what you're looking at. It's just one cell layer lining the tube, but you are looking at two sides of a tube. And how would I know that those are not two cell layers? Simply remember, if they were two cell layers, you wouldn't have this space in between. I don't have this space between cell layers. Epithelium is supposed to have closely attached cells, closely packed cells. All right, so what is the shape of the cell? So how many cells should I be looking at a time in order to tell what is the shape of the cell? Look just at one cell at a time. One cell at a time. One cell at a time. Which one do I look like? Epithelium that is squamous, cuboidal, or columnar? Just look at one cell at a time. So this is the height of the cell. This is the width of the cell. So the cell here is much longer than it is wide. So this is gonna be a columnar cell. So what type of epithelium am I looking at in here? I'm looking at an epithelium that has one layer of tall cells. So one layer, simple. Tall cells, columnar. So this is my simple columnar epithelium. Simple columnar epithelium. All right, so let's check this one after break. We can